Life, as you well know, is an energy-intensive process. It takes energy to operate muscles, extract wastes, make new cells, let the brain process and send messages, and even, from time to time, do some serious thinking. An organism's entire energy budget is spent on the cell level. In some cells, as much as half of a cell's energy output is used to transfer molecules across the cell membrane, a process called active transport. Cell movements require energy, and thousands of energy-hungry chemical reactions go on in every living cell, every second, every day. The kind of energy cells use is chemical bond energy, the shared electrons that hold atoms together in molecules. Most cell processes use the same energy source, the rechargeable energy carrier, adenosine triphosphate. ATP has this arrangement, a molecular unit of adenosine coupled to a chain of 3 phosphate groups, thus the name, adenosine triphosphate. The phosphate groups are held to each other by very high energy chemical bonds. But under certain conditions, the end phosphate can break away, and the energy released used for energy-hungry reactions that keep a cell alive. When the end phosphate is released, what is left is ADP, adenosine diphosphate. This change from tri to di is taking place constantly as ATPs circulate through cells. The recharging of ADP to ATP requires a huge energy investment, and that energy comes from the food we eat. How energy is extracted from food molecules and used to synthesize ATP is one of the great discoveries of modern biology. All nucleated cells contain mitochondria, the tiny bodies where ATP is produced. This energetic protist is literally filled with them. But the best way to see them clearly is to squeeze the cell under a cover glass until it ruptures, forming a thin membrane bubble, a little aquarium in which float a few of its mitochondria. A mitochondrion consists of 2 sacs made of membrane. Folds in the inner sac increase the surface area for chemical reactions that produce ATP. By breaking up mitochondria and separating out the membranes, biochemists have discovered exactly where the chemical reactions involved in synthesizing ATP actually occur. First, mitochondria take in molecules derived from food, molecules with lots of chemical bond energy, the breakdown products of sugars and fats. Sugar contains enough chemical bond energy to burn with a hot blue flame. Fat contains even more. Fat has about twice the energy content of sugar. If you can boil 1 test tube of water on a spoonful of sugar, a spoonful of fat will boil too. To understand how energy is extracted from these fuel molecules, we need to climb right inside of a mitochondria. In the space between the 2 sacs, fuel molecules are disassembled in a way that releases their chemical bond energy. This energy, in the form of electrons, drives molecular pumps embedded in the inner membrane. The pumps push hydrogen ions obtained from the fuel molecules into the inner membrane sac. It's like blowing up a balloon. It's during this process that oxygen plays its role. Oxygen has a powerful attraction for electrons. Think of the electrons released from fuel molecules as a stream of water. What oxygen does is lower the stream bed dramatically, so that the water, the electrons, can do a lot more work. Some bacteria can live in oxygen-free environments, but also have the ability to use oxygen if it is available. Without oxygen, the cell can make 2 ATP molecules for every sugar molecule metabolized. With oxygen, the same cell can make 36 ATPs from each sugar molecule. Oxygen's powerful pull on electrons allows most of the energy in their fuel molecules to be used to push in the hydrogen ions. The folded inner membrane is studded with enzymes. These enzymes, ATP synthase, offer an opening through which hydrogen ions can escape. As they exit through ATP synthase, they generate the energy required to bond the terminal phosphate onto ADP, converting it to ATP. That's it. That's how the ATP battery is recharged. But what happens to all those carbon atoms that originally made up the fuel molecules? In the process, they combine with oxygen to form CO2, carbon dioxide. Carbon dioxide leaves the mitochondria and escapes through the cell membrane, where it's picked up by the bloodstream and transferred to your lungs, ready for a good exhale. That's animal respiration. Oxygen in, burn fuel molecules, make ATP, carbon dioxide out. But how about plants? For them, it's carbon dioxide in, oxygen out. Most of us hear about photosynthesis around grade 3 or 4, when our teacher explained that plants take in carbon dioxide, light shines on them, they give off oxygen and make sugar, which is then turned into starch or some other carbohydrate, and now you know why. Photosynthesis is the way plants make fuel molecules to feed their mitochondria. In terms of getting energy, the only real difference between plants and animals is that plants make their own fuel molecules, whereas we animals, in order to get fuel for our mitochondria, must eat our green benefactors, or eat something that has. Under the microscope, a leaf cell looks like a box full of green jelly beans. These are chloroplasts, bodies containing chlorophyll molecules. Chlorophyll is a unique substance that absorbs the energetic blue and red wavelengths of light while reflecting away green. The first electron micrographs of sections through chloroplasts stunned biologists. These were definitely something more than little green jelly beans. They were bodies with an elaborate internal structure. Chloroplasts contain stacks of hollow discs called thylakoids. And the thylakoid discs are covered by a carpet of chlorophyll molecules. In these green carpets, light energy is converted into chemical energy, a process that drives the living world. When light hits a plant, some of the light energy absorbed by its chloroplasts is used to split water molecules into hydrogen ions and oxygen. The oxygen, a waste product, enters the atmosphere. The hydrogen ions are used in making ATP, the same energy carrier produced by mitochondria. And this is how they do it. When a chlorophyll molecule absorbs a photon of light energy, it transfers the energy to an electron. So the arrays of chlorophyll act like solar panels, producing electron energy. But instead of flowing down a wire, the electrons flow through molecular pumps that pump hydrogen ions into thylakoid space. Just as in mitochondria, these pressurized ions pass through enzymes that create ATP and other energy carriers. These molecules supply the energy for the food-making reactions of photosynthesis. Food-making chemistry occurs in the soupy fluid that surrounds the thylakoid discs. Here, in a complex series of reactions known as the Calvin cycle, carbon dioxide is coupled with longer carbon chains using energy supplied by ATP and other energy carriers acquired from the light reactions. This cycle produces a kind of molecule with a rather intimidating name of phosphoglyceraldehyde, PGAL for short. Some of the PGAL molecules keep the cycle rolling, and some of the 3-carbon PGALs enter an enzyme that unites them to form the 6-carbon sugar glucose. That's what your 3rd grade teacher was talking about. Carbon dioxide in, light on, out comes oxygen and glucose sugar. So, the cell's 2 energy-transforming organelles, mitochondria and chloroplasts, feed on the waste products of each other. CO2, given off by mitochondria, is exactly what chloroplasts need to make PGAL, the building block of sugars and other carbohydrates. And the oxygen released by chloroplasts during the light reactions is exactly what mitochondria need to drive the electrons that pump in the hydrogen ions, making it possible for ATP synthase to add that terminal phosphate to ADP, creating ATP, the universal energy carrier. And that's how cells get energy.